Good morning, good afternoon, and good evening. We thank you all for joining us today for the webinar. My name is Tim Devling, and I am Divisional Director for Life Science here at XStrahl. It is with pleasure that I am joined by Nick Stout, the CTO from Smart Scientific Solutions, and by Frank Verhagen, co-founder of Smart and professor at the University of Maastricht. Nick is going to present on another highly innovative tool developed by Smart Scientific Solutions, which addresses the need for better dosimetry and reporting for cabinet irradiators. Before I hand over to Nick, just to let you know, the webinar is being recorded and a link to view it again will be sent out after the broadcast. If you have any questions, feel free to type them in the chat box and we will discuss at the end. So without further ado, over to you, Nick, and radiation dose calculation software for non-image guided irradiators.
Thank you, Tim, for the introduction. First, I'm going to give a brief overview of what we're going to discuss today during the talk. For those who don't know us, I'll introduce Smart Scientific Solutions. Then I'll talk about the current state of dosimetry in radiobiology, followed by a few papers. One is a discussion about the transition from cesium-137 to X-rays. Then I'll talk about the methodology of how the dose is usually calculated and the reporting. After that, I'll have a look at what is feasible when doing hand calculations without any advanced tools. And then I'll finally introduce our software solution, SmartRad. What is it and how does it work? What changes in your workflow? And I'll mostly do this through demonstrations with recorded clips of the software in action. And finally, I'll also discuss the validation we did at Oxford University. So about us, we're a company that was founded in 2015 in Maastricht, in the Netherlands, and we focus on developing products that support preclinical radiation research. We also have very strong relationships with the academic world. We have 3 centers within walking distance of our office, and we also collaborate quite often with them. Some of our products you might know. So we create phantoms. Left, you can see the plastic creatures with a rat and a mouse phantom. And on the right, you can see the micro CT phantom that is used as a QA and calibration phantom for image-guided irradiators. Our most known product is our preclinical treatment planning system, Smart XPS. With this system, the user can do advanced beam configurations, calculate them using Monte Carlo dose calculations, evaluate the dose, and when content with the plan, he can send this instructions to the SARP. So now I'll go over the current state of the radiation research without image guidance. So a lot of those irradiators that are still used for these type of studies are cesium-137 irradiators, but there is a gradual transition to X-rays. This is because many governing bodies are pushing towards replacing the cesium irradiators and also maybe in the future ban them. The reason for this is because the powder in the cesium irradiators could theoretically be used to create a dirty bomb. So there's a safety issue. Over the course of many years already, many machines have been replaced, and there's more than 1,500 machines already using X-rays. But this is not without challenges. So you go from a monoenergetic high energy photon beam, or almost monoenergetic, to an X-ray spectrum. The X-ray spectrum has a lot more low energy photons, resulting in a very heterogeneous distribution and more sensitive to tissue changes or medium changes, making it harder to calculate the dose. Currently, most dose calculations are done in a point dose fashion. So people calculate by hand what the dose rate is at a point dose, and this is the dose that gets reported. We can also state that dosimetry reporting is very important to provide reproducible studies, and we'll discuss that more later. So I'll use a few papers that were published relatively recently. The first is about inter-institute variability. So that's a study by Peterson et al. And he sent 6 acrylic cylinders with TLDs to 12 different institutes. And 5 of those institutes have an X-ray tube. The other 7 had a radionuclide source. And they were just requested to give a 1 gray dose to 3 of the cylinders and a 4 gray dose to 3 of the other cylinders. And what you can already see here is that while there is some variation, it is clear that the X-ray sites are performing worse and have more variation, already showing that it's much more difficult to calculate the dose for X-rays. So the current dosimetry practice is calculating the dose rate at a prescription point. This is usually based on a measurement at a calibration point, and then corrected for the distance using the inverse square law. When done correctly, of course, there's additional corrections. One of those corrections is depth, to take into account the attenuation through the specimen, and backscatter corrections to also give a correct dose rate for the material behind the scoring point. There's a paper by Zhong from 2020 that looks at the accuracy of this prescription point method versus measurements and Monte Carlo. So they themselves applied all these corrections to a prescription point and then did ion chamber measurements and Monte Carlo simulations on different geometries. So solid water blocks, a small water phantom, a mouse phantom, a rat phantom, and then a mouse in a pie cage. What we can see here is that there's a very large deviation of 15 to 37% for the different geometries, but between the measurement and the Monte Carlo simulations, agreement is within a few percentage. And one important thing to notice is that these phantoms, while having a different geometry, are still homogeneous. So it's just one single material. So in theory, in reality, the deviations are going to be much larger. As mentioned before, dosimetry reporting is very important for the reproducibility of studies. In a paper by Draeger et al. In 2020, it was reported that overall there is a lack of dosimetry reporting. They analyzed more than 1,700 papers and scored sites, each paper, on what they reported. You can see that they report dose, source, and these parameters quite often. But things like dosimetry protocol, medium, backscatter factors, they're often forgotten. It is also not a quality issue because with higher impact factor of the journal, this did not improve. In one of the figures, you could even see that it slightly decreased as the impact factor improved. This, however, is not new. In an earlier paper from 2023, so 7 years earlier, the National Institute of Standard Technology published a paper that came with approximately the same results on a different data set. So now we had a bit of literature. I'm going to have a quick look at how the hand calculations would compare to, for example, a Monte Carlo simulation. As mentioned before, the depth dose correction is one of the important factors, but this assumes a very simple geometry. There's no different media supported. You have one medium and you determine the attenuation through this media. For example, if you come from a cesium irradiator and you would take a big block of water, as shown here, you see a gradient, but nothing extreme. And you could, in theory, calculate at a certain depth what the dose is going to be there. Because cesium has a high mean energy of 609 keV, you can expect a very minimal gradient. When you look at X-rays, the gradient increases, but it's still very possible for such a simple geometry to just calculate by hand what the dose at a certain depth is going to be. As you decrease filtration, the exact depth becomes more important, but still, as mentioned, you could do this analytically. However, depth dose correction is not the only factor. You have backscatter. How this is done usually is with lookup tables, and they have a limited range of setups. The backscatter is dependent on a range of factors, namely energy spectrum, beam size, material. And here I show a simple example of some backscatter factors that would not really be tabulated normally with 80 kV copper filtration and a 60 by 60 field. You can see that for a 5 centimeter thickness PMMA, you get a backscatter factor of 1.17. And for steel, it will be 1.04. Often forgotten is that, for example, low energy photons and steel will actually cause less backscatter because of the high photoelectric cross sections. But your setup probably does not look like a simple block of water. So a more realistic setup would be, for example, a mouse. Here you can see the same spectra being used on a mouse. On the top, you can see the cesium-137, and you can still see a homogenic phantom. It has a gradient. You see a bit more dose to the bone, but you could probably still approximate this with a point dose. But once you go to the X-rays, what you see is because of the high amount of low energy photons always present in the spectrum, is that you get an extremely high bone dose and also a shadow cast by the bone in lower regions. And it becomes almost impossible to calculate how this distribution would be by hand. And for the middle one, that's a realistic spectrum to be used. I think you can't really approximate that anymore with a point. The bottom one, it becomes completely impossible, but I would never advise to use a spectrum that does not have an external filter and only the inherent filtration. So now I finally get to introduce the new product. So as mentioned before, it is a dose calculation software for fixed radiation cabinets called SmartRad. What we will be supporting is the CIX1, 2, and 3. And it is focused to be easy to use. So it is aimed at biologists who don't need a large amount of physics knowledge or Monte Carlo knowledge. This is all handled by the software itself, but it does use the gold standard calculation method, namely Monte Carlo simulations. It provides the user with advanced tools for dose evaluation, such as 3D dose through color washes, as you saw earlier already, but also things like dose volume histograms and metrics to analyze certain structures. It's focused on flexibility. So we want to allow the user to create a range of setups depending on what kind of experiment they want to run. And we don't want them to be limited with what the tool can do. So you can use a range of shielding conditions, holders, and it's very flexible in use. We do provide a lot of standard specimen that you could use, but you can also modify them. And it works on a large range of photon energies and filtrations. It is intended to be used on a calibrated spectrum, but it can, at the cost of slight bit more uncertainty, still calculate dose outside of the calibrated range for the photon spectrum with internal scaling factors. So you can do virtual experiments if you're thinking about using a completely new spectrum. And very importantly, it can also export a dosimetry report, so you'll never have to fear that you forgot one of the advised parameters that NIST wants you to report. So how does the workflow look? I'll quickly go over it, but I'll show it in clips. So first you would select your irradiator. Then you would build your experimental setup. I will be showing a cell culture and the mouse with a lead shield. Then you evaluate the dose from this specific setup. And if you're happy with the settings, you can export your dose report and report everything like this in your paper. And there are also a few advanced features, such as evaluating X-ray spectrum changes in case you are interested in this for a study, or modify existing or create your own geometries from scratch, things like specimen, but also shielders, fixation materials, those things. So here you can see the setup module from SmartRad. On the left, you can see the 3D display showing how your virtual setup looks like, and this should mimic your experimental setup. On the right, you can see all the changes you can do to a specimen, a shield, a holder. So it's basically your virtual setup. And on the bottom, you can change things about your irradiator. So like your shelf height, your tube current, and also a few parameters regarding the simulation itself. I'll start the video and I'll create a cell culture. So here you can see, you can select the specimen type. For cell cultures, we support well plates. By default, it's a 96 well plate, but you can still modify the number or parameters, like for example, the wall thickness. I'll adjust it to have a slightly thicker wall. Aside from that, you can also tell how high certain layers are going to be. So for example, this is a 1 millimeter thick cell layer, probably a bit too thick to be realistic. And then you have a different shelf height, and you just start the calculation. You get a pop-up regarding the process of your calculation. Here the clip already stops. And what's important to know as well is, like on the bottom right, you can see, for example, the simulation mode. You have a fast one where you can quickly prototype if you're still wondering what kind of experiment you want to do, what kind of energy you want to use. Then you can do a full one, which has very good statistics. Or if you're an expert user, you can use the custom mode and adjust all types of Monte Carlo parameters. Then after your simulation is finished, you can go to the evaluation mode, where you will see on the left the 3D, the displays of the slices. And on the right, you can see some structures. For this case, for example, the specimen are the cell layers. And you can select that so you can see some parameters there. We can also show a, here we'll scroll through it for a bit so you can see how your wells look like. You can also look in 3D to have an idea how it is all positioned in your irradiator. And on the plant side, you can choose the beam on time, or you can set like, for example, a mean dose to the specimen, which in this case is the cell cultures. You can also look at the metrics, like the dose tables. For example, the mean dose here is approximately 10 gray. You can add different dose metrics that are commonly used in radiotherapy, like the D95, the D5, things like that. I'll also quickly still show that you can really see the medium difference in kilovolt X-rays by zooming in on the well. So you can see that you have like the PMMA wall and the soft tissue, or in this case, water, I think, in this layer. And you can see the dose difference between just PMMA and water already. Once you're finished with that, you can go to the export module, where on the left, for the beta version, it's not in the user interface yet, but on the left, you can normally get options to export things like the specimen you used, the shielding you used, and you can share it with different SmartRad users. On the right, you can see the dosimetry report. So this is all the information that will be available in your paper. So the information about the radiation source, such as the spectrum characteristics, the field size, the geometry, in this case, like it's a well plate. Also mentions what kind of table it was using, what material the table was from, some parameters regarding the simulation, the dose scoring, it's dose to medium and medium here, but also about your dosimetry protocol, which is also important. And of course, it's not only in the user interface you can see it, but you can also export it as a text file or an HTML file, after which it will look something like this, an editable text format. This is not from the same case. And you can incorporate this in your papers. Then to the irradiator selection. I kept this last because it combines some of the advanced features. Of course, you have the simple, just selecting which irradiator you're going to work with. And usually you will also have to choose a tube potential. But here you can see that you can adjust, for example, the tube potential. You can adjust the filtration and instantly look what it does to the energy spectrum, like the distribution. And at the bottom, you can even see what changes with the characteristics, like the half value layers, the mean energy, those parameters. If needed, you can also export this spectrum for analysis in another toolkit, or maybe sharing it in your paper if there was something evaluating multiple spectra. The second advanced feature is the creation of your own geometry object. I will cut this a bit short because otherwise it would take too long. But I'll show how to, for example, extend the mouse. So we pick a starting geometry here, just some default shaped mouse. SmartRad will convert it to an editable format. And you can see on the top a 3D display, on the bottom a 2D display, and you can start modifying these geometries as you wish. So here we're going to add the mouse tumor just by adding a simple sphere. You can obviously change viewing direction, so you can position the mouse and the sphere as you like. As you can see here, it's just a soft tissue tumor that we add here. It's not really important that it's very correct. When you're happy with your shape, there's a bunch of operators you can do. Here, we basically combine the shapes, but we keep the original tissue, so only the region outside gets replaced. You can do it the other way around, replace all the regions. You can also use shapes to cut pieces from your geometry. That's, for example, when you want to use the geometry editor to create a shield, which is what we did with the next setup. So the next setup, we show a custom geometry, which we created earlier. I did increase the size of the tumor so it's visible here. And we also pick a shielding. Here the shield is very large circular cutout. And yeah, just for demonstration purposes, we also incline the shield a bit to show that you can pretty much make very strange shapes if you would like. Once you would start the irradiation with this and it finishes, you would get the evaluation panel as shown previously with the cell cultures. But because it's a more complex geometry, some of the features become more interesting. We have the same features, so you can see in 3D where you're located. You can look at the color maps, so just the color wash, but you can also select structures. What structures you can see is something you can change a bit in the settings. There are a lot of available structures. Some are a bit too small. Here, for example, we'll take the soft tissue, the bone, and also the tumor you just added. And that can be used to evaluate metrics. I'll also hide the structure so you can still see the color wash quite nicely. About the color wash, you can also pick different color washes if you want to see gradients. Some other color maps are usually a bit better, like Jet is very good to emphasize gradients. But because we have this complex geometry, it makes sense to use things like the dose volume histograms, where you can see what percentage of your region of interest is getting a certain dose. Here you can see that, for example, the bone is actually getting a higher dose at some points, but on average, a lot of it is behind the shield, causing it to be less mean dose. That sort of shows what the SmartRAD is capable of, but it's nice that it can do all that, but it's also important that it is accurate. For this, we collaborated with Mark Hill's team from Oxford University. They did a series of measurements. So one of them is depth dose measurements at different spectra. So they did at 100 kV, 200, and 300 kV, all with the available filter of 0.2 millimeter copper. This was important to test if SmartRAD could scale between different spectra. In Monte Carlo, it's typical that you only have one calibration factor for one specific spectrum, and then when you want to use a new one, you have to calibrate again. SmartRAD can go outside of that region, but of course, we did have to test this. Then we also tried two different materials, plastic water and bone. That was tested for spatial agreement. This was done using different shielding sizes and positions. And then also the radial distribution was evaluated. And hopefully soon it will be published in Physics in Medicine and Biology. It is in the last phase of the review. I will show a segment of the depth dose measurements. Showing all the results is a bit too much for this presentation. But for example, on the right, you can see the figure for the plastic water. The full line with the color wash around it is the Monte Carlo simulation. And then the box with the error bars are the measurements. You can see that everything is within uncertainty of each other, which is very good, of course. The maximum deviation we saw was 1.9% with a mean of 0.8. This is important to know that it was calibrated for one energy, 300 if I recall correctly, but it scales well to the other energies as well, even though it uses the same calibration factor, correcting internally. For RB2 bone, we saw slightly bigger differences, what is to be expected because you have the bone equivalent material, making it a bit more difficult to match correctly, but the mean was still only 1.8% difference, and there were only two measurement points above 6.1%, above 5%, with the maximum being 6.1%. Then for the spatial agreement, we use multiple shields, so 3, 5, and 10 millimeter. This was done to see how well we would represent this beam size. And it's placed on top of the stack of solid water, and then we still take a depth, several depth measurements. It was also irradiated at two positions, the central axis, as shown in the picture, and then an exact 10 centimeter offset to the lateral side. The results of this also showed good agreement. It's a bit harder to see on the figure, but the solid lines are the Monte Carlo simulations from SmartRAD, and the dots are the measurement points on the film. It's hard to see, but that actually makes it good because that means they're overlapping very well. The maximum deviation on all the things we did, so with lateral shifts to 3 different field sizes, we had a 0.44 millimeter maximum deviation, and on average, the mean average is 0.24 millimeter. This is sub-millimeter accuracy, which should be sufficient for non-image guided applications. We assume that the slight deviations are usually caused by differences in positioning, because you don't have a real reference point, and the voxelization within SmartRAD, because when you're combining all those geometries and giving it to the Monte Carlo engine, he needs to pick a resolution, which here it was 0.3 millimeter, and you get slight shifts in positions, but acceptable in this case. I will not go further in all the other results, but you're free to read the paper once it's published in Physics in Medicine and Biology. I thank you all for listening. And before I give the word to Tim again, I want to briefly mention that in April in Antwerp, there's a 6th conference on small animal precision image guided radiotherapy. And as far as I know, registrations are still open. So I hope to see some of you there. Thank you.
So we'll just wait. So really appreciate that presentation. That was really, really interesting. We have quite a few questions that have started coming in as you've been speaking. So what I will say to everybody is if you do want to ask a question, you can type it into the little question box and we will start asking Nick and Frank. So thanks for being here today, guys. I'll start from the top. Is an obvious question. How long does it take to calculate dose?
Well, that's an obvious question, but a simple one. So it depends a bit on what you want. The fast mode, which is more intended for prototyping, so like quickly checking what your dose distribution would do with different setups, that's only a few seconds because you're allowing for larger uncertainties and you're also taking coarser voxels. For a full Monte Carlo simulation, typically, again, it depends a lot on your setup, but it's a few minutes. Unless you're interested in like out of field dose, then you need to up the time a bit more to get good statistics.
What variation do you see in kind of dose delivered to a mouse as calculated by SmartRad as compared to, for example, the beam on time you would expect just using an in-air calculation?
So you mean like the differences in dose that we calculate compared to if you would do that point calculation?
Yeah.
Well, we didn't do point calculations ourselves. We don't have a machine yet, but that's back from in the presentation. There was Zhang et al. Who had a paper that said around 30% difference for a homogeneous mouse. So I would assume that it's probably even more.
Okay, so it could be quite large.
Could be quite large, and it's also dependent on what you're interested in. Is it the dose to the bone, or is it the dose to soft tissue, and where is this located?
Okay, interesting one. The computational mouse phantom that you've shown, does that come with the software, or will users need to provide their own?
No, so SmartRad has a database of pre-generated mice and rats, and also a few other geometries. But the software that was used to generate those is Moby and Roby from Duke University. So some people might be familiar with it.
Yeah, and if I may add, it's about 6,000 pre-generated rodents.
Yeah, different sizes. And you can modify them yourself with the geometry editor if you want this plank tumors, things like that.
So now we're getting into the devil in the detail. A few of the, you might guess who some of these questions are coming from, but which detectors were used for the PDD measurements in the different materials?
EBT3 film.
So film-based.
Yeah. And we also had the ion chamber measurements for the air karma, obviously.
Okay, and what solid water did you use?
Plastic water LR was the solid water, and the bone material was RB2.
Okay, and then going kind of to the biology of this, quite actually an experimental setup is it possible to directly import a CAD drawing of shielding, for example?
At this time, not yet. So you have, because I don't know if with CAD you could theoretically, but if it's, for example, an STL, you would have no idea what the material inside is. So we start from a raw geometry, like a voxelized geometry in SmartRad itself. And at this time, you have to create it in SmartRad. So you can't import a CAD yet. I don't exclude it for the future.
So for at the moment, you can draw the shape, you can draw, you'd have to do it yourself by hand, I guess. Interesting question. You've shown an example using a 96 well plate. Can you also simulate tubes, for example? So I guess Falcon tubes or other types of container.
Yeah, so in theory, you can simulate any geometry you can make with basic geometric shapes if you create it yourself in the geometry editor. The 96 well plate one is just a standardized well plate that we know where we had like technical drawings from, but you can in theory also do others. But the culture tab that generates the well plates is an analytically generated geometry. But if you would make a custom geometry of those tubes, I'm not too familiar with how the all the different cell culture geometries look like, but you can make them with the geometry designer and then import them.
Okay, so essentially it could potentially be quite flexible. So if you are using, you know, other sorts of lab plasticware, you could make those shapes.
Yes.
And I guess as we develop this, as we learn what people want to do, we can start making those shapes and using them as a.
Yeah, so we can also provide them in advance to people.
There is a question about whether or not this would be suitable for patients. I think that's kind of a bigger discussion. So I think I will mention it because it was asked, but I think that's kind of a broader topic. But keeping with the mice and keeping with the cells, is there a limit to the dose rate that you can use here? So would it work on Flash?
So, well, again, so there's not really a limit in dose rates that you could use. There is a limit in the X-ray potential that we support. It's from around 30 kV to 500 kV. So that's what we model the spectrum for. Dose rates, in theory, wouldn't matter, but there are no advanced tools to evaluate dose rate yet. But those things could be added in the future as well. Like, for example, you get histograms based on dose rates, thresholds, and things like that. But they're not in there at this time. It's not intended yet for Flash. But interesting.
Yeah, that would be whether or not there's a dose rate effect. Probably not, but an interesting question. Quite an easy one for you now, Nick. For the cell culture radiations, can you adjust the medium or the level of medium in each well?
Yeah.
I think that is a
you
yes.
can adjust the media from the plates, the walls, the cell, and then the material you use to like fill it out, basically.
Okay, so you can use. Do you have water? I guess you have water, maybe.
Yeah, in this example, I had the cells as soft tissue, then a layer of water above it, and then the plate was PMMA. But you can change those materials to I think there's around 18 or 20 different materials, but it's relatively easy to extend that as we get user feedback.
There's a interesting, so lots of interesting questions. Sorry, Nick, coming in. How frequently would we need to do a dosimetry calibration, or would the system need to be recalibrated?
So
now,
the answer would be it would need to be recalibrated if you replace your X-ray tube, or if as your X-ray tube ages and you get a different dose rate with the same settings, then you would have to recalibrate. So it depends a bit on how your QA is, but the calibration is based on the dose rate that your X-ray tube outputs. If that dose rate changes, you obviously need to recalibrate SmartRad. So
I mean, we know the tubes on the 6 systems are very, very stable for a number of years, but I think we would always recommend, you know, a reasonable quality assurance time scale. It doesn't change, but you know, you want to make sure it doesn't change. But I guess, yeah, Nick, as you say, if everything stays the same, the software should not need to be recalibrated either. So if you use a 96 well plate and you cover half the plate with a lead shield, do you think it would be accurate enough to calculate the dose delivered to the cells or the cell chambers underneath the shielded part of the plate?
Yeah, so that's one thing. So in theory, you can do that. At this time, we don't support the shields on top of the well plates yet. The well plates are, to get good resolution, they're a bit more restricted in what you can add from shielding and things like that. We will add that later. But one thing you always have to take into account with Monte Carlo simulations in general is that out of field, those calculations, which if you put a shield above the well plate, would be the case, is that you need a lot more particles to get good statistics under that shield. So it would be accurate enough if your simulation parameters are correct, but you would have to simulate for a much longer time because you're not just simulating the main beam, you're just only simulating a bit of scatter. So the simulation time would increase a lot, but it should be possible.
Okay, and moving to mice, can the software calculate dosimetry for multiple mice? Which is a very nice question.
Yeah, at this time, we are still looking into that. So there are some limitations in resolution when using the Monte Carlo engine. So in theory, you could have like a custom geometry again, where you have multiple mice, but you have to take into account that because it's getting voxelized into something that fits your computer memory for a lot of arrays, you will probably have very coarse resolution compared to a single mouse.
Okay, so I guess, I mean, so the answer will be yes with a but, which will be improved. I can imagine people will want to use, you know, a pie cage for TBI with potentially multiple animals, and it's nice that we'll eventually, or we can, you know, to some degree, resolution dependent, achieve that now. Another lovely question, which is, can I try it? And I like that sort of question. Thanks for asking it.
That's a good question. So we are currently going to release the beta very soon. Then we have a few dedicated sites that will be testing it. And then after that, shortly after that, hopefully the commercial release will be there. And then you can always contact Tim, I guess, to schedule a demo.
I would be more than happy to schedule anybody with a demonstration of this. It is a really good tool, and I think we can do the demonstration quite well over Teams or remotely as well. So yeah, so please get in touch with us after this. One question that has just come up, and it is it's quite a nice one, is does this meet reporting requirements? And I think that corresponds quite nicely with a recent publication of what those requirements should be. So Nick, does it meet reporting requirements?
So the version I showed you used the recommendations from Draeger et al. And the paper from NIST. And I saw indeed, I think it was in November that the other paper was published with the new requirements. We still have to go through and see if we meet every requirement, but we will make sure that everything that the software can know will be reported. Because I know that in the newest paper there was one thing about, did you do an in-experiment measurement? Things like that will obviously not be in there because the software can never know that, but things about dosimetry and dose distributions and things, that will all be in there. And we will obviously also follow literature. If new recommendations come, we'll try our best to keep up to date.
I think we'll be continually evolving this this software over the next couple of years and so on. So it's a really nice tool. I mean, it's a very novel tool as well. So we'll be developing this and adding to it as people, I guess, make requests and so on. And I think we're coming up to 20 minutes of questions. And I've got one final question, which I'd probably like to end on because I do like this question, is when do you think it will be available?
It's always hard to say a strict deadline, but okay, yeah. So the beta is in a few weeks, and then we have a short period where the beta testers are actually giving us feedback regarding the software, and depending on how many things we still need to change, it'll be hopefully within a few months, by summer approximately, something like that. Then commercial versions will be released normally.
Oh, brilliant. So that's great. So thanks a lot, Nick, for an excellent presentation. Lots of interest from this. Everybody who's on the webinar will receive, or who registered for the webinar will receive a link to the video. We'll also contact you as well. If you've got any further questions, then please feel free to get in touch with myself, and I can arrange for Nick or Frank to answer those. And just to say a big round of applause again to Nick and to Frank for coming up with a wonderful idea and for a great presentation. And I wish you all a good beginning or a good end of your day. So thank you all.